Welcome students. I am R.K. Pradhan, Department of Botany, Khalikot Higher Secondary School, Barampur. Last class we discussed about the structure and function of cell membrane and also some introduction part of nucleus. But today I will discuss about the detailed structure, function, origin of nucleus. First we have to know what is nucleus or what are the definition of nucleus, distribution of nucleus, position of nucleus, number, origin of nucleus. One by one I will discuss. First we have to know what is nucleus. Nucleus is the largest component of the cell. Nucleus is the reproductive organelle of the cell. Nucleus is the autonomic organelle of the cell. Nucleus is the controller or regulator of the cell because it controls all the vital activities of the cell. So, nucleus is called as controller or regulator of the cell, just like we used in our home the regulator of fan. We also regulate the fan by rotating the regulator, like the nucleus is a regulator or controller of the cell because that controls the metabolism or vital metabolic activities of the cell. So, that is also called as control tower of the cell, regulator of cell, autonomic organelle of the cell. These are the different functions performed by the nucleus. Then come to the discovery of nucleus. First, Robert Brown, you know, Robert Brown discovered the nucleus from the orchid root cell. Orchid root cell. Robert Brown discovered the nucleus from the orchid root cell. Then their distribution, we know nucleus are commonly found in eukaryotic cell. Commonly found in eukaryotic cell, but absent in prokaryotic cell, as well as RBC, matured mammalian RBC, and sieve tube of phloem. These are the generally they are found commonly found in the eukaryotic cell, but absent in prokaryotic cell, matured mammalian RBC, and sieve tube of phloem. These are the distribution of the nucleus. Then come to the origin of nucleus. What is the origin? Every nucleus originated from a pre-existing nucleus. That is given, this concept given by E. Strasburger. E. Strasburger has reported that every nucleus originated from a pre-existing nucleus. We also know the process of karyokinesis, nucleus divide and to form a daughter nuclei. So, this is the concept given by E. Strasburger. He reported that every nucleus is originated from a pre-existing nucleus in the process of karyokinesis. Karyokinesis means division of nucleus into two daughter nuclei. That process is called as karyokinesis. Then comes to the number of the nucleus. A single cell may contain a single nucleus. That cell is called as monokaryotic cell. Monokaryotic cell or uninucleate cell. Uninucleate cell. Those cells which contains a single nucleus, that is called as monokaryotic cell or uninucleate cell. Most of the cells having uninucleate cells. Then second type is dikaryotic cell. Dikaryotic cell or binucleate cell. Binucleate cell. What is binucleate cell or dikaryotic cell? In those cells having two nuclei, that is called as dikaryotic cell or binucleate cell. We know the Paramecium caudatum is a protozoa which is an unicellular protozoa which contains two types of nucleus. One is micronucleus, another is a macronucleus. Macro means large nucleus, micro means small nucleus. Micronucleus is reproductive function, macronucleus controls the vegetative function of the cell or somatic function of the cell. But micronucleus divides by mitotic cell division or mitosis, whereas macronucleus divides by amitosis method. Okay. Then third type cell is a polykaryotic cell. Polykaryotic cell or multinucleate cell. Polykaryotic cell or multinucleate cell means those cells having more than two nuclei. That is called as a polykaryotic cell or multinucleate cell. We know these polykaryotic and multinucleate cells are again two types. One is sinusotic condition. Another is a syncytium condition. What is sinusotic condition? In sinusotic condition, the nucleus divides repeated karyokinesis that form a large number of nuclei, but without cytokinesis. That condition is called as sinusotic condition or multinucleate condition. You remember, a single nucleus divides by repeated karyokinesis or nuclear division, but without cytokinesis, without cytoplasm division. That form many nuclei inside the cell. That condition is called as sinusotic condition or multinucleate condition. This is most commonly found in fungus like mucor and rhizopus. Then another condition is syncytium condition. Here, in syncytium condition, the nucleus divides four nuclei without nuclear fusion, but cytoplasm fusion. Here, four nuclei remain inside the cell, but here the cytoplasm undergoes fusion, but the nuclei never fuse with each other. That condition is called as a syncytium, especially that is found in bone marrow, where syncytium or multinucleate condition is found. This is the number of nucleus present in different group of organism. Then comes to the position of nucleus. Generally, nucleus is located at the geometrical center of the cell. Geometrical center of the cell. Generally, position of the, if we go to the position of the nucleus in different type of cell, we will find generally the nuclei are located at the geometrical center of the cell. Then in case of adipose tissue, where the nuclei are located at the periphery of the adipose cell or adipose tissue. You remember, nuclei are located at the peripheral portion of the adipose tissue. Similarly, we go to the spirogyra. Spirogyra, which is a filamentous alga which contains a nucleus inside a vacuole which is suspended by cytoplasmic strand. You remember, in case of filamentous alga like Spirogyra, nucleus remain inside the vacuole which is suspended by cytoplasmic strands. Similarly, in case of yeast, nucleus may be present inside the vacuole or that may be separately present outside of the vacuole. In case of yeast, nucleus may be present inside the vacuole, that is called as vacuolar nucleus, may be present outside of the vacuole. Okay, these are the different position of nucleus in different group of organism. Then comes to the structure. Then another thing is acetabularia. You know, acetabularia is a largest blue-green alga which has a basal cell or rhizopodial cell. Rhizopodial cell contains a nucleus at the basal part of the cell, but the apical cell does not contain nucleus. Here, according to the scientist Hammerling experiment, if the upper part of the acetabularia cell breaks, then the part will be degenerated. Only those part of the cell which contains nucleus, they can able to exist in the earth surface, exist in the different earth surface. But according to this concept, here the nucleus is only located at the basal or rhizopodial region of the acetabularia cell. Okay, these are the different position of the nuclei found in different group of organism. Then comes to the structure of nucleus. What are the structure? How the different part of the nucleus perform different function? We will discuss one by one. Then come to the structure of the nucleus. Here, generally nucleus has structurally nucleus has four components. One is nuclear membrane, second is nucleoplasm, third is nucleolus, fourth is chromatin network. First we will discuss about the nuclear membrane. Let us see the diagram. This is called as nuclear membrane. This is outer nuclear membrane, then inner nuclear membrane, then perinuclear space. Then this is called as this is endoplasmic reticulum. This is called as annulus. This is called as nuclear pore. This is chromatin network. This is called as nucleoplasm. Nucleoplasm. This is called as nucleolus. Nucleolus. Here, the nucleus consists of 4 components. One is nuclear membrane, second is the nucleoplasm, third is the nucleolus, fourth is the chromatin network. You remember, first you see the nuclear membrane of the nucleus. The nucleus is covered by a covering that is called as nuclear membrane. The nuclear membrane is also called as karyotheca. Study of a nucleus called as karyology. Similarly, the nuclear membrane is also called as karyotheca. The nuclear membrane has 2 covering. One is outer nuclear membrane. Second is inner nuclear membrane. Okay, the space between outer and inner nuclear membrane, that is called as perinuclear space. The thickness is about 9 to 10 nanometer. The thickness of perinuclear space is about 9 to 10 nanometer. Similarly, the nuclear membrane is also perforated at several places by small opening, that is called as nuclear pore. The nuclear membrane is perforated by several small pores, several small openings, that is called as nuclear pore. The nuclear pore is a octagonal shape which contains a octagonal protein, that is called as annulus. You remember, annulus is a octagonal cylindrical protein structure, that is called as annulus. That helps in regulation of closing and opening of nuclear pore. What are the function of nuclear pore? Nuclear pore help in exchange of materials between the nucleoplasm and cytoplasm. After the synthesis of ribosome or RNA or protein, they are transported from the nucleoplasm to the cytoplasm through nuclear pore. This nuclear pore is octagonal shaped, whose diameter is about 20 to 80 nanometer. That is octagonal shape. And annulus is also a cylindrical shape structure whose diameter is about 20 to 80 nanometer. Annulus is only responsible for regulating the opening and closing of the nuclear pore. That during the closing time, that plug the nuclear pore, but when the different material exit from the nucleoplasm to the cytoplasm, during that time annulus opens. That means that helps in function as a regulator of opening and closing of nuclear pore. Okay, this is all about your nuclear membrane. Then second part is the nucleoplasm. Another thing is the outer nuclear membrane is continuous with the endoplasmic reticulum. Means that is connected with the endoplasmic reticulum, but inner nuclear membrane encloses the nucleoplasm. Then second part is the nucleoplasm. What is nucleoplasm? The nuclear membrane encloses the viscous colloidal protoplasmic substance, that is called as a nucleoplasm. This is a colorless substance, so its name is karyolymph. Karyolymph, you know lymph. Lymph is a colorless fluid connective tissue, just like the nucleoplasm is colorless, so its name is karyolymph. Karyo means nucleus, lymph means colorless fluid. So nuclear membrane encloses the colorless colloidal proteinous substance or colloidal protoplasmic substance, so its name is karyolymph. But what are the function of nucleoplasm? Nucleoplasm contains different substance like DNA, like RNA, like proteins, and DNA polymerase enzyme, RNA polymerase, and ADP, ATP, ADP, and AMP, adenosine triphosphate, adenosine monophosphate, and some nucleosides. These are the nucleoside or nucleotide which present inside the nucleoplasm. Basically that contain DNA deoxyribonucleic acid, RNA, then protein and DNA polymerase enzyme which helps in DNA replication, RNA polymerase enzyme which helps in transcription, then different nucleoside and nucleotide which are present inside the nucleoplasm, that is ATP, ADP, and AMP, adenosine monophosphate. But actually the nucleoplasm lack microfilament. Nucleoplasm lack microfilaments. I have already said in the cytoplasm of eukaryotic cell contain microfilament, so cytoplasm has cyclosis movement, autonomic streaming movement of cytoplasm. But here in nucleoplasm no microfilaments are present. They never help in they don't perform the cyclosis movement of the nucleoplasm. So what are the function of nucleoplasm? Nucleoplasm is only responsible for different nuclear reaction, where DNA synthesis takes place, where transcription takes place, where different protein synthesis takes place. That is the seat for different nuclear reaction. Nucleoplasm is a seat for different nuclear reaction. Then comes to the third component, that is nucleolus. We know nucleolus is just a small spherical acidophilic body. Small spherical acidophilic body which remain attached at the secondary construction of chromosome during cell division. But nucleolus is a membraneless body of the nucleus. It is without membrane. It has no membrane. Nucleolus is a small spherical body which without membrane, membraneless body of the nucleus. This is the component of the nucleus. So who discovered the nucleolus? Fontana discovered the nucleolus. Fontana. First you know what is nucleolus. Nucleolus is a small spherical shaped acidophilic body which remain attached at the secondary construction of chromosome during cell division. Fontana first discovered or observed the nucleolus. We know nucleolus is only found in eukaryotic nucleus but absent in prokaryotic nucleus. Okay. Then nucleolus has a three component. A nucleolus has a three component. One is suppose this is a nucleolus. That is called as chromatin, peripheral chromatin. This is the peripheral chromatin. This is called as a pars amorpha. This is called as pars fibrulosa. This is called as pars granulosa. Here the nucleolus has three main components. One is pars amorpha, second is pars granulosa, third is pars fibrulosa. the nucleus is covered by a peripheral chromatin material. This is covered by peripheral chromatin material, but it has no membrane at all. So it is a membraneless body of the nucleus. It is only covered by a peripheral chromatin material. Then inside the chromatin material, three components are present. One is pars amorpha, amorphous matrix, means the ground substance of the nucleolus is a proteinous substance where the protein and a RNA ratio is 2:1. You remember, inside the nucleolus the protein and RNA ratio is 2:1, means 2 part is protein and 1 part is RNA. So this is the amorphous substance, amorphous proteinous ground substance. So this part is called as pars amorpha. Then another part is amorphous, that is also called as amorphous matrix. Second part is granular region or pars granulosa. In this region, different granular proteins are present. Ribonucleoproteins are present, or RNA ribonucleoproteins are present, whose diameter is about 150 to 200 angstrom. 150 to 200 angstrom. Its name is also called as pars granulosa. Pars granulosa. Then another part is pars fibrillar region, that is also called as pars fibrulosa. Here the protein fibers are fiber-like structure, thin thread-like structure, whose diameter is about 50 to 80 angstrom. Whose diameter is about 50 to 80 angstrom. So its name is pars fibrulosa. One thing you remember, the pars granulosa, here nucleolus is only responsible for synthesis of ribosomal subunit. Ribosomal subunits like a 60S ribosomal subunit and 40S ribosomal subunit. These ribosomal subunits are generally produced from the pars granulosa. So sometime question will ask, which is the precursor of ribosomal subunit? Pars granulosa is the precursor of ribosomal subunit, means ribosomal subunits are made from granular region or pars granulosa. Similarly, sometime question will ask, which is the precursor of pars granulosa? That is pars fibrulosa, means fibrillar region is meant for formation of granular region. Granular region is meant for formation of ribosomal subunit. Okay, these are the different three main part of the nucleolus. Then what are the function of nucleolus? Nucleolus is only responsible for synthesis of ribosomal subunit as well as RNA synthesis as well as a different protein synthesis. I have already discussed the ratio of protein and RNA in the nucleolus is 2:1. 2 part is protein and 1 part is RNA. So three main function is associated with the nucleolus. One is the synthesis of ribosomal subunit, second is synthesis of protein, third is synthesis of RNA. Okay, these are different function of the nucleolus. Then comes to the chromatin network. We know chromatin network are the fourth component of the nucleus. First component is nuclear membrane, karyotheca. Second component is nucleoplasm, karyolimb. Third component is nucleolus. Fourth component is chromatin network. But actually during when the cells remain in the non-dividing state, the chromatin material appeared in the form of thin filamentous and network structure. Thin filamentous and network structure. In the non-dividing state of the cell, chromatin material remains in the thin filamentous and network structure. So that is called as chromatin network. This chromatin network discovered by E. Strasburger, first discovered by E. Strasburger. But when the cell convert into dividing state or division state, the chromatin network gradually condensed, shortened and thickened, that form a structure that is called as chromosome. You remember chromosome? During the process of cell division, you know interphase and karyokinesis. In the interphase state, chromatin materials are appeared, but when the cell goes to the karyokinesis phase, the chromosomes are found in the different phase of the karyokinesis. But actually the chromatin materials changes into chromosome. Again we will discuss about the chromosome. Chromatin material discovered by E. Strasburger. Then after Waldier discovered the chromosome. Waldier. Waldier discovered the chromosome, E. Strasburger discovered the chromatin network. Okay. You know chromosome is the physical unit of heredity, or chromosome is the hereditary material of the cell that is responsible for transmission of parental character from one generation to next generation. So this is a physical unit of the heredity, or that is also considered as a hereditary material of the cell. Then come to the first we have to know the discovery of chromosome discovery. Waldier discovered the chromosome. You remember Waldier discovered the chromosome, but the chromatin material discovered by E. Strasburger. I have already said chromatin found in the non-dividing state of the cell or in the interphase in the form of thin, filamentous and network structure. But when the cell convert into dividing state, the chromatin network changes into condensed, thickened and coil-like structure that is called as chromosome. Chromosome discovered by Waldier. Then come to the size of the chromosome. Size of the chromosome. We have already known the size of chromosome is always greater than the plant chromosome size is greater than the animal chromosome size. Similarly, the monocot chromosome is greater than the dicot chromosome. Dicot chromosome, most of the birds having smallest chromosome. Birds having smallest chromosome, longest chromosome found in trillium. You remember trillium, whose chromosome is the longest chromosome. Plant chromosome is longer than the animal chromosome. Monocotyledon plant chromosome is longer than the chromosome of a dicotyledon plant. This is the size of the chromosome. Then comes to the shape of the chromosome. Shape of the chromosome. On the basis of position of centromere, the shape of chromosome is of different type. First one is acentric chromosome. First one is acentric chromosome. Acentric means centromere is completely absent in the chromosome. That looks like a rod-shaped structure. A rod-shaped structure. Then second is a telocentric type of chromosome. Telocentric type of chromosome, when the centromere is located at the terminal end of the chromosome, that looks like a I-shaped structure. So that is called as a telocentric chromosome. But in case of human being, telocentric chromosome are completely absent. In human being, telocentric chromosome are completely absent. Then acrocentric chromosome means chromosome is centromere is located just behind the terminal end of chromosome. Centromere is located just behind the terminal end of chromosome. That is called as acrocentric chromosome. That is also called as a J-shaped chromosome. Then another is a submetacentric chromosome. That is a L-shaped structure. Means the centromere is located just behind the middle part of chromosome, that forming a short arm and a long arm. That looks like a L-shaped structure. But in case of acrocentric chromosome, the centromere is located just behind the terminal end of chromosome, forming a shorter arm and a longer arm. That looks like J-shaped structure. Sometimes question will ask, a chromosome with a shorter arm and a longer arm is called as dash. That is acrocentric chromosome or J-shaped chromosome. Sometimes question will ask if a chromosome looks like a L-shaped structure, that is called as submetacentric chromosome, or L-shaped chromosome is called as submetacentric chromosome. Then another chromosome type is the metacentric chromosome. The metacentric chromosome is a V-shaped structure. Means the centromere is located at the middle part of chromosome. V-shaped chromosome, so its name is a V-shaped structure. But actually we have to know the acrocentric chromosome is specially found in different chromosome of human being, that is the 13, 14, 15, 21, 22 and 21 chromosome. You remember 13 acrocentric chromosome found in 13, 14, 15, 21, 22 and Y chromosome. But we have 23 pair of chromosome, 46 number of chromosome. But acrocentric chromosome is only found in 13th chromosome, 14th chromosome, 15th chromosome, 21st chromosome, 22 chromosome and Y chromosome. You remember it. Then second type, submetacentric chromosome. Submetacentric chromosome is specially found in 2nd chromosome number, 4th chromosome number, 5th chromosome, 6th chromosome up to 12th chromosome number. Then 17th number of chromosome, 17th chromosome, 18th chromosome and X chromosome. Submetacentric chromosome, you remember, submetacentric chromosome found in X chromosome, acrocentric chromosome found in Y chromosome. Okay. Then metacentric chromosome are a first number of chromosome, third number of chromosome, 16, 19 and 20 number of chromosome. These are the different type of chromosome shape found in different number of chromosome in human being, from first chromosome up to the 22 number of chromosome. You remember only basic Y chromosome is the acrocentric chromosome, S chromosome is the submetacentric chromosome. This is the shape, size discovery. Then comes to the number of chromosome. You know number of chromosome. One is diploid number of chromosome. Diploid means two set of chromosome present in a nucleus. That is called as a diploid number of chromosome. Diploid number of chromosome found in somatic cell, and germ cell, reproductive cell. Okay. Then second thing is haploid number of chromosome. Haploid means one set of chromosome, a complete one set of chromosome, that is called as haploid number of chromosome. Haploid number of chromosome found in gametes, reproductive units. But in case of reproductive cell or germ cell, the diploid number of chromosome convert into haploid number of chromosome. So in gametes, male gamete or female gamete, that is sperm or ovum, where haploid number of chromosome present. In case of meiosis, cell division takes place in reproductive cell or germ cell. In germ cell, in meiosis cell division, the diploid number of chromosome changed into haploid number of chromosome. The diploid number of chromosome reduced to half number of chromosome. One set of chromosome, complete set of chromosome present in a gametes. Another things you know, genome. Genome means a complete set of chromosome, that is called as a genome. Study of a genome, that is known as genomics. Okay. These are the different number of chromosome. Regarding the number of chromosome, then comes to the structure of chromosome. Structure of chromosome. Look at the picture. Here this is called as a chromatid. This is called as chromatid. This is called as a chromonema. Chromonema. This is a small bead-like structure present on the chromonema, that is called as chromomere. Chromomere. This is called as centromere. This is called as kinetochore. Kinetochore. This is called as pellicle. This is called as matrix. This is called as a nuclear organizing region. NOR, nuclear organizing region. This is also called as a secondary construction. Secondary construction. This is called as telomere. Telomere. This is called as SAT chromosome. SAT chromosome or trabant. That is also called as trabant. You remember? Then comes to the structure of the chromosome. First, we just give an idea about. First, you know DNA. DNA give rise to nucleosome. Nucleosome give rise to solenoid. Solenoid. Solenoid give rise to chromatin. Chromatin loop. Chromatin loop give rise to chromatid. Chromatid give rise to chromosome. This is the structure of the chromosome. DNA, diameter of DNA is about to 2 nanometer. Nucleosome, whose diameter is about 10 nanometer. 1 nanometer is equal to 10 angstrom. Solenoid is about to 30 nanometer. chromatin network is about to 300 nanometer, chromatid is about to 700 nanometer, chromosome having 1400 nanometer. You just give a skeletal framework of genetic material or hereditary material. First DNA, then nucleosome, then solenoid, then chromatin, then chromatid, then chromosome. Here you look at the picture. Here a metacentric chromosome. Here this is a metacentric chromosome, means those chromosomes present in the metaphase, but the membrane of chromosome is called as pellicle. Chromosome is covered by a covering that is called as pellicle. Pellicle encloses the ground substance that is called as matrix. A metacentric chromosome consists of two symmetrical strands. This symmetrical strand is called as chromatid. Chromatid is the unit of chromosome. Chromatid is the half of the chromosome. A chromatid contains two chromonema. Chromatid contains two chromonema. Chromonemata plural form, chromonema singular form. Inside the chromatid contains a thin filamentous structure that is called as chromonema. Chromonema, or that is also called as a DNA. The chromonema contains many small bead-like structure that is called as chromomere. Recent cytologists reported that chromomere are regarded as a gene. Then next part is the centromere. Centromere is the, or that is also called as primary construction. This is the first narrow part of the chromosome. This part is also called as a centromere that determines the shape of the chromosome. On the basis of location of a centromere in the chromosome, that determines the different shape of the chromosome, like V-shaped chromosome, like L-shaped chromosome, like J-shaped chromosome, like I-shaped chromosome. Similarly, outside of the centromere, some proteins are present that is called as kinetochore. A disc-shaped protein which remain attached on the centromere, that is called as kinetochore. Kinetochore is responsible for synthesis of a microtubule. You remember, kinetochore is responsible for synthesis of microtubule. Similarly, the second part is, another part is secondary construction. This is the second narrow part of the chromosome, that is called as secondary construction, where there is a origin or point is present, that is called as nuclear organizing region. In this region, nucleolus is formed. Chromosome is responsible for formation of nucleolus. In this region, nucleolus is originated. Then another, the last terminal part of the chromosome, that is called as SAT chromosome or trabant. SAT means sign acido-thymonucleochromosome. Thymonucleochromosome. At least one SAT chromosome present in a genome. You know genome? Genome is a one set of, complete set of chromosome called as genome. At least one SAT chromosome present in a genome. The SAT chromosome is only responsible for formation of a nucleolus, and the terminal end of the chromosome, that is called as telomere. Terminal end of chromosome, that is called as telomere. Then another two things are here there are two types of chromatin found in the chromosome. One is heterochromatin region, another is euchromatin region. Heterochromatin region is specially found at the telomere region, centromere region, and nuclear organizing region. You remember 3 point only. Heterochromatin region are found only 3 region at centromere region, telomere region, and nuclear organizing region. But in other part of the chromosome, euchromatin region is present. But what is the basic difference between heterochromatin region and euchromatin region? In heterochromatin region, these regions are dark region, means they are condensed region, they are compactly packed with each other. So this is dark region. When this region is stained with some chemical that absorb more color, so that becomes dark. Heterochromatin region are dark, but euchromatin region are light region, means they are non-condensed, they are not less condensed. But heterochromatin region is not those genes are present, they are not actively perform the transcription. Active transcription is only seen in euchromatin region, means those genes which are present in euchromatin region, they perform active transcription. Transcription means the process in which RNA synthesized from the DNA, that is called as transcription. In euchromatin region, active transcription takes place. In heterochromatin region, inactive transcription takes place. And this is also, this is non-condensed, less condensed region of the chromosome, but heterochromatin region is the condensed region of the chromosome. This is the basic difference between heterochromatin and euchromatin region. But what are the function of chromosome? Chromosome, the main function of chromosome is the transmission of hereditary character from one generation to another generation. But in case of some lamb-brush chromosome, which also responsible for synthesis of yolk in some organism like insect, but actually chromosome is the physical unit of heredity. This is your last part of the cell structure and function. In the next class, I will discuss about the biochemistry or biomolecules like carbohydrate, protein, fats, amino acid, nucleic acid. Okay, thanks for watching this video.